Hi everyone, this is Jason, producer of Data and Biotech. Before we get started, I wanted to let you know about our latest white paper. It's a comprehensive guide to implementing machine learning models in biotech manufacturing. It's a complete overview of all the potential problems of ML adoption and, more importantly, how to solve them. To download it, simply visit connect.cordine.com/biotech-ML. We've also dropped the link in the show notes of this episode. Okay, let's get into it. Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they are using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we sat down with Pierre Salvy and Lucile Bonnin from Cambrium, developers of NovaColl, the first micromolecular and skin-identical collagen designed specifically for highly efficacious skincare formulations. During the interview, we discussed the process of protein design, specifically touching on the role of ML models across this process, the challenges of integrating data in the lab, and the development of a protein programming language. Lucile and Pierre also unpack data digitalization in the lab, protein design and vector databases, designing the manufacturing process, and de-risking quality control in scale-ups. Here we go.
Pierre Salvy and Lucile Bonnin, welcome to the Data and Biotech Podcast.
Thank you, Ross. Nice to meet you.
Nice to meet you too. And just to kick us off, could you each give us an introduction to your career to date? Lucile, why don't you go first?
Yeah, sure. Hi, Ross. Thank you so much for the invitation. So I'm a chemist originally coming from France, from the West Coast, now based in Berlin. I worked in big company before in personal care and food and beverage industry before doing my PhD in physical analytical chemistry. I developed there like a chemical platform for stable hair colorations. That was really fun with a lot of scales and big tanks. I then jumped into predictive analytics for chemistry, working in lab for Henkel, which brought me to basically think about data in a different way, which I brought to my R&D kind of background, research and development background. And from this point on, I decided never again to do R&D without using data. I think you have tools in your life and you just should use them. So I brought them to consulting for a while, and now I'm working for Cambrium, where basically the synergy of R&D and data is perfect. And I'm leading the R&D team here at Cambrium for 3 years now.
Awesome. Pierre?
And Pierre, thank you again for the invite. Super happy to be talking about the exciting stuff we do here at Cambrium today. Historically, I'm a big fat nerd. I studied engineering, applying math to a bunch of different domains, from material science to microeconomics to nuclear physics. I actually almost got a degree in nuclear physics. But then I went to the US West Coast, decided to make jet fuel by engineering genetically beyond yeast. That was really fun. And I decided I really like this, biotech. So I got a PhD in the field. And then the founders of Cambrium found me and Lucile. And that's basically the startup, when we built the company that we're going to tell you about.
Very cool. And so tell me about Cambrium. What's the company's mission, and how does it accomplish it?
What we do is we use generative AI to design proteins that will be basically the building block of the sustainable biomaterials of tomorrow. The general idea is most of the stuff that surrounds us, right, like the table on which this computer is, the computer in itself, it's based on a lot of unsustainable material, right? Coming from street mines, also from animal exploitation and so on. And there are actually a lot of material problems that have been solved by nature for the last 2 billion years of evolution, right? And we'd be a missed opportunity not to, take from these really nice materials that, make up your hair, your skin, your bones, and try to find applications to that, right? I think that was the original idea.
Yeah. So it's a biomaterials company. You're focused on, developing sustainable biomaterials. My understanding is that you have a biomaterial that you've already begun to bring to market. Is that right? Can you talk a little bit about what that is?
Yeah. So NovaColl is our first product. It's a micromolecular collagen. So what does that mean? It's basically a vegan collagen. So a lot of application in the personal care and beauty care industry comes from animals, unfortunately. So it's not cool for the animals and also absolutely not sustainable. So our first product is basically tackling this issue. Pipeline is developing in other direction, but we launched last year. That was super exciting. So it means that for back in the day, 3 years company, we already were at scale, at industrial scale, to be able to commercialize our first product, which I think for biotech companies is pretty impressive, but you have to be that fast nowadays, to keep market traction.
So from idea to selling, it took less than 2 years, which is absolutely unheard of in synthetic biology. But also, what was the status quo before, right? How do you get collagen before? And collagen was a byproduct of the meat industry, right? So your cow goes to the slaughterhouse, gets slaughtered, and then you have a lot of remainders. Then basically what people were doing is boiling this, extracting the collagen there. And and then selling that to ingredient manufacturers, and then we put them on creams, right? So like people were literally putting dead animals on their skin. And the moment you tell them, they're like, oh, well, that's gross. I'd rather buy the vegan collagen, which is good for us, right? And that's basically also how we chose this initial product. Is like the market traction was already there so strong that for us, we were like, wow, that's a no-brainer. That has to be our first product. And I'd like to talk also a little bit about why cosmetics, right? Because with each material science, we sell cosmetic. First, your skin is a very complicated material. But second, cosmetics is also a great market to enter when you're a tiny company, right? Because it's low volume, high price elasticity, high drive for innovation, and a lot of permeability of the market for basically new players, right? Not the case in healthcare, not the case in commodities. So in that respect, it's really a place to start for many companies, I would say.
As I heard the story of NovaColl, one of the things I was wondering was, did you start out with the idea that Cambrium was going to enter the cosmetics industry and that collagen was like a right target to replace with biomaterials? Or did you develop sort of the data and software platform to explore the protein space and then select collagen and the cosmetic space and go from there? So how did the decision to use this as your first target evolve?
I think every targets we've been addressing were pushed very hard and also in a very logical framework by the business development first. But the pipeline that we used was, in terms of data, was already very abundant, right? From what we could do, especially for the personal care industry, right? So what Pierre is developing with all his data platform is very oriented to create the product and design it in specificity. But when it comes to business, this is actually something where we got in touch with customers very, very early. This was early days, right? Because now we are building a data which is proprietary to us with our own lab. So it's becoming richer and we have more freedom into actually guiding the business development into the right use cases. So it becomes more and more interesting.
Yeah, it was kind of concurrent, right? Like so we screened around 300 use cases. So protein market fits, if you are, which involved incredible conversations, but basically with people that were actually welding a rocket in the garage somewhere. So that's really cool. But also, at one point, you're going to build a protein, right? And so you know what are going to be the building blocks you need in your data infrastructure, but also like your general software infrastructure. We're going to talk about the toolkit that we built, right? Whatever protein you're going to make, there are some common elements, right? So these common elements we were already building at the time. And then once we decided to go for collagen, there were like some more specific elements that we just mentioned for that.
So since I'm certain many of our listeners are sort of new to protein design, it would be useful if we could sort of talk about the problem at a high level. You have an idea of a target market that you'd like to enter with, a protein that you could potentially design. What are sort of the components of the process or the different problems that need to be solved between knowing what market you want to get into and, designing that first protein that can actually solve the problem that you're setting out to solve?
I'm going to use the space glue example because we're not going to make space glue, so I'm not infringing on any IP. But basically, you're talking to people and they're telling you, so I have these two materials, I want them to glue together, but I also want it to be sustainable because in space, if something burns, you're in a can, contain air, it's toxic for everyone, not happy, right? What's nice about proteins, like if it burns, it just smells like burnt hair. It smells bad, but it's not going to kill you, right? So there was a real use case for it. And so basically, people tell us, okay, we want these material properties, for example, being able to stick metal to plastic, and we want it to be curing in so many minutes at that temperature. We have databases that allow us to check, what are similar use cases in nature. Turns out the mussel, like the seafood, really good at making a water-resistant glue to glue on the rocks, right? And this is a protein glue. So basically what we will do is look at this protein sequence, identify the part that is responsible for gluing, and basically adapts the rest of the protein to a particular use case, like lighting temperatures, curing time, and so on. This adaptation is a small work for many methods that we have that span mechanistic engineering to AI, right, or generative AI. But basically, that's how we start. Basically, from a functional element that we know that is what characterized, and then basically we shift around. And then basically, the design space there is often gigantic. So for NovaColl, the design space was around 10 to the 18, right? It's just big. We do really smart sampling and screening selection with computers. And we basically down select to a bunch of high likelihood candidates. And we select the top 10% and then basically turn that into a DNA sequence, order the DNA inside the lab, and then basically pass it on to Lucile.
And then it goes to my lab, to the wet lab, which is a beautiful place. And it's incredible that you can basically screen there because it's already de-risking the work I'm doing together with my team. Once the DNA arrives, you basically clone it into an organism. We call them our microfactories. So this is basically what is generating the molecule of interest. We screen them according to different factors. You want to check if your organism is actually secreting what you think it's secreting. So that's the first step. Then you want it to be very productive. It's great if it's producing the molecule that you want, but if it's a very low productivity, not going to be able to commercialize that. And then you want to check its material properties. So everything there is by Pierre's team. But yeah, you want to check that the safety and the efficacy is actually aligned with your commercial usage. So in the terms of base glue, is that actually sticking under which shear rates, etc. So we're checking everything in the lab in high throughputs. We can also talk about automation later because this is something we onboarded very early on, because even though we de-risk the use case by having very few candidates coming to the lab, it's still a lot of candidates, right? So you can't run that many things manually. So right now we are at a rate where actually we can screen more than 1,000 candidates a week with our weekly platform, which I think is pretty cool and outstanding.
Yeah, that's fantastic. So it sounds like the business development team, like when they're first talking to these potential customers about potential applications, needs a menu of all of the different properties of proteins that they might select from. And so that, Pierre, you can then take that menu and then select the candidates. Am I thinking about that right? It just seems like a very complex ecosystem where the communication would be difficult.
Yeah, I mean, of course, communication is key, right? And the truth is, you have information exchanges in both directions. Sometimes we had use cases where it's like downright impossible, like people that wanted something that's water resistant, but also biodegradable in a salt bath. And you're just like, well, if it's going to rain on it, then it's biodegrading. So like sometimes you need to align this, right? Because actually the truth is often the customer doesn't really know what they want. They're like, I want something sustainable. I don't know how it translates, right?
I think on NovaColl, there is a really cool example. So we try to bridge the gap between sustainability and efficacy. So for an anti-aging like NovaColl, our first product, we bring anti-aging properties. No surprise, but you want to be as good as what is on the market. Then on top of that, you want to offer new things which were not there before. So we have a peptide, we identify specific peptides, which is also antioxidant, which is the reason number one why your skin is actually aging. It's like oxidative damage. And the molecule is doing both at the same time. So I think it's really interesting to see that on top of like leveraging what exists already, you're also bringing new things to the market, to this new technology. So that's pretty exciting.
It is very exciting and very interesting. It's kind of starting from the beginning of developing the data systems that you needed to explore the protein landscape and understand the different features of the proteins that were out there so that you could identify the candidates, you could subset them, leading up to handing it off to Lucile's team to, do the experimentation. Pierre, can you just walk us through what the data journey was like? How did you build up those systems? What were the different components of the systems that you were developing during that early phase?
Yeah, I want to be very honest from the beginning. It's like, it wasn't something that just built from the get-go, right? And worked. So it was just a bunch of scripts and a bunch of like clever implementation of like sampling algorithms. I think that may be like 3 different important milestones through this journey. One was, if you're going to screen, you're going to get data. If you get data, what can you infer from it? And how do you include this inference loop inside your design, right? So one of the first things we did was actually simulate the whole lab in a computer to understand what were the data input outputs, what were also the possible throughputs with the money we had, right? And basically from this, we're like, okay, this is the data that we can use to improve our algorithms at the later stage. Same time, we developed a little bit the protein design toolkit, which took the form of a protein programming language at the time. So what we realized is that the way people talk about proteins is very functional. It's like, oh, this bit is responsible for that, right? But believe me or not, at that point, there was no tool to do that well. People were literally copy-pasting protein sequences in Word files. And I heard this horror story as somebody getting their words fine autocorrected, for spelling, which changed the protein sequence without them knowing. It took them 6 months to debug why the thing wasn't working. So anyways, that was the real thing, So we're like, okay, if you have a functional description of something that's letter-based, then maybe you can just write a programming language, right? That was before the LLM boom. And so we did this. We wrote our protein programming language, which allowed us to actually design a lot of stuff in a very smart way. If it's a proper language that's compiled and so on, this will be ingested by our AI to basically do diversification and optimization of the sequences. And that's basically how we design NovaColl. And of course, then the LLM boom happened, and the way we looked at protein sequences, data changed a bit. Now we actually have, like pretty much everyone probably, we have our own RAI for, gathering information from my experiments and basically for me, the next design, the next experiments. We have diffusion model for proteins. Everything was added ad hoc to our little garden of models. And today, I think we have one of the nicest gardens.
I want to sort of unpack some of the components of what you said a little bit. So when you say building, a model that acts as your lab, that's the model that does the experimental screening for you to determine what the characteristics of a given protein sequence is, or what does that mean to you?
I think it's closer to what people market as a lab digital twin now. So not necessarily simulating what's happening in the experiments, but simulating the lab operations, right? Like, so what are the hubs of data? What are the failing points? What are the throughputs? So that you get an idea of like, are you going to get a gigabyte of data per week, per day, per month? What type of data? And basically also which machines you need to buy and what are the marginal costs you need to pay in order to increase your throughput and so on, right? So that's really basic. But if you don't do that, then you buy the wrong machines, then you lose 6 months and you can't launch in 2 years.
Right. So it's a lab optimization algorithm to determine what are the different components of the lab that you need in order to execute end-to-end the entire screening and manufacturing process that you need.
Exactly.
And maybe jumping on that, you have to imagine that when you work in a lab, you have a lot of machines which are not supposed to work together, right? So you're going to have like a shaker, an oven, like a machine which is all going to screen your proteins, and then they don't communicate together. And I think one of the base work of doing that is like really understanding your lab end to end and put all those pieces together to have like the right database afterwards. That was a huge work that we started with Pierre in the beginning. So basically designing the lab, right, buying the right machines. You have to imagine also R&D is not always, the most innovative place because you work with very expensive machine. And sometimes some of them, they're not able to communicate with like a standard computer. So there were a lot of like crazy challenges that we faced in the beginning we were not even expecting. But now it's all connected.
You have to imagine the vendor calls with like the people that sold us the robots. Like Lucile was asking all those super sharp questions and the guys were like, okay, I think I can deal with that. And then I was hanging in the back like, yeah, but does it have an API? And just like, they're like, what do you mean? And I would start pitching them. I have an AI in the cloud. I want you to control the machine. Is that possible? And then literally some of us told us, why would you do this? Mm-hmm. And other person who told us, oh, but you have an iPad and there's like a button on the iPad and you can tell your technical assistant via the AI how to turn the button. I'm like, I don't think you understand the vision, but that's cool.
Yeah, that's very funny. And, that's been a running topic of conversation on this podcast as well about, the way that these different automation tools don't interface with each other and sort of the challenges of creating the data that you need to understand what's happening in your lab by integrating all of the different data sets together is unnecessarily challenging. It's difficult to do ML and AI if you don't even have the basic data infrastructure in place and data integration in place to, run the models on.
Absolutely.
So Pierre, back to the protein programming language that you had and sort of the way that LLMs and diffusion models and more of the recent advances in ML and AI have changed things for you. I'm curious, what were sort of your intuitions that led you to the approach of the protein programming language, and what are these new models doing for you that's sort of, I'm assuming, replacing the need for that?
When I was working on the West Coast, so I was working at Amiris with Total New Energies who are making this biofuel, right? And so there they had this genotype specification language that they could use. And I was actually really sick. They had a bunch of DNA banks and you could just type in ligation between the DNAs and build your own DNA construct, hit enter, it would compile it into like robotic instructions and build the DNA. So you would have basically almost DNA as a service, which is absolutely insane. We are not trying to repeat exactly the same thing, but the use case was the same, right? You have sequences, you have functional bits within the sequence. There is a way to actually arrange it in a functional way that is not relying on like fuzzy natural language, but is actually, computer instructions. So literally you would say like, hey, here I want sticky bits. So you would go to the database, take one of the sticky bits, optimize for whatever I want, then add a linker, then here I want the blocky bits, and so on and so forth, right? And once we are able to like write that on a piece of paper, it made a lot of sense. And we just got to implementation there. And I think it was super cool. But then LLM came in and it kind of overpowered it a little bit. So now we have tools that allow us to specify natural language and basically influence diffusion generation so that basically we'll have the protein that we want. It's not a one-to-one replacement, but it allows you to have basically more structure-based elements taken into account rather than sequence-based, which is important for some types of proteins. But the answer is we use a bit of everything.
Yeah, so that's interesting. So the protein programming language is sort of a combinatorial approach where you have these different elements that have functions that you've seen in the wild associated with these portions of DNA, and then you combine them to select a set of the most likely candidates and then combine them together. And then those are sort of the sequences that then go out to be ordered and go through Lucile's lab to determine whether they're going to work. But these more powerful models, because they've been trained on such a large corpus of data, they have, sort of a deeper understanding of the structure of the proteins and the functions of the proteins. And so I'm assuming they can get you closer to the right protein faster because they sort of have that understanding already. Am I thinking about that right? Or are there pieces I'm missing?
It's just yet another way to explore the forest of possibilities, right? It's like, we had sonar and now you have a LIDAR on top, right? And you have pros and cons for both. So protein programming language, for instance, you get what you type, what you optimize for, right? At the end of the day, as it said, it's basically a combinatorial multi-objective optimization problem, which marketing doesn't like me saying this because it sounds too nerdy, but at the end of the day, that's what it is, and I think it's exciting. It doesn't hallucinate. Your deep network might hallucinate, and you have very little control on this. So we use a bit of both.
Yeah, that makes a lot of sense. So as you're moving toward maturity, what has the development of the data platform looked like? Has it been moving to the cloud and, doing more parallelism, using Kubernetes? How's that journey looked for you?
Yeah, I think we analyzed really the R&D pipeline and where we can leverage data. And I think we identified 3 fields which were very, very useful. First one, we addressed it deeply now already, which is protein design. Then we looked into strain engineering. So there is plenty of application, which are super interesting. Pierre is working on to try to optimize how our manufacturers are actually working. This was also from a strategical perspective, one focus to have. And Pierre, I think, wrote a PhD on that, so he was pretty qualified for the job. And then we looked into manufacturing and scale up. As a chemist, if you look into scaling up processes, it's where you just going to meet the most problems. It's crazy. That's where you put the most money, and you really don't want to miss on that. So strategically, it also made a lot of sense to look into scale up. So really early on, we integrated all the bioreactors that we have in the lab, for example, with Pierre, to make sure that all the data through all the process is actually uploaded to the cloud, and that we are also able to kind of correlate that with our partner who is producing for us at large scale. And now we have this kind of correlation that we have a 1 liter in the lab, which is correlated to how exactly work a 1 ton at our partner who is producing for us. And we are able to tweak the process with that. And this was very obvious because we put a lot of money there, but you also have to, for the sake of speed and competitiveness, to be kind of really fast over there. So that was a very cool angle to go for. Yeah, and not still continuing working on those 3 axes, actually.
And so to put that in the context of the specifics of your question, Ross, from the beginning, we were cloud native. So I like to say digital native, but from the beginning, there were no notebooks, no local files, nothing. Everything went to the cloud from day 0. And that's what allowed us also to move so fast forward, because first you store your data, then you try to organize it. But it's always cheaper than 10 years later trying to organize the data that is in unstructured format, right? Ask the BASF or Bayer of this world how much money they're spending on trying to retrofit an infrastructure on their existing data pipelines or data, notebooks. Terrible. So from day 0, we are cloud native. Then what has changed is, of course, the technologies we've been using. So for instance, now that we're doing more and more protein embeddings, for instance, we're starting to use vector databases, we're benchmarking several solutions. Of course, we have a lot of structured storage. We're starting to move towards a little bit more of unstructured data storage, testing different methods and solutions there as well. And for that, the AI research assistant we built, the RAG basically, was a good way to beta test several features.
In the beginning, you were sort of designing the manufacturing process from scratch. And so if you could just talk a little bit about how you set a vision for what the manufacturing process should look like, and any insights you want to bring from your PhD are welcome as well.
Yeah, I think manufacturing. So the same way we actually mapped the lab, we mapped kind of our supply chain, right? So you have all the R&D which is happening in the lab, and you scale it up to a certain scale. Unfortunately, we don't have massive fermenters in the lab yet, but we have a lot, and it does the job. And then you look into a partner we produce with a different partner who are covering those steps at large scale. You obviously don't get to the lab to manufacturing at commercial scale like in one day. You have what you call a scale-up phase, which is pretty impressive and also very data heavy. So here the engineering team from Pierre helped us a lot to try to understand how would we de-risk the tech transfer. So you really have, in terms, when you run a precision fermentation, you really have to understand what's the input, what's the output, what's the risk, and how do I read the data to make the right decisions during production. So this was like kind of a collaboration between both teams. Yeah, and then you ferment, and then you filter everything, you clarify everything, and then you stabilize it and make it like commercializable. So we actually have 2 partners in place for that, which is very also rich in data that we also collect on our ELN to make sure that this is transferable as we scale.
I would like to point out 2 things that are really not obvious, but actually fantastic in what Lucile just said. One is, if you ferment 10 cubic meters or 10,000 liters, that's 10 tons of material, right? That's a gigantic amount of things to process. The second thing is having correlation between the fermentation in a 1 liter tank, right? This is the size of a water bottle, and the 10 tons tank is insane. That's what scale-up is. It's like you go across 4 orders of magnitudes and you get exactly the same cake you're trying to bake, right? And This is something, if you've done a little bit of biomanufacturing, is absolutely amazing because it takes years for other processes to get there.
Yeah. And that's kind of what I did during my PhD when I worked on the chemical platform. So you realize that the physical law of which are in action in the big tank are actually completely different than in a small glass tank in the lab. And you have to transfer basically those law from a mini scale to a very large one and try to understand how to de-risk that. And every time you work in a different setup, basically you have to recalibrate your model. So that's what we did. And that's why it's also important to have a very good partner in the first place and not to work with everyone because, you want this database to be consistent and also to be super qualitative. We scaled up from lab to a very high scale, I think, in 6 months, which brought us to commercial scale. When you would have told me that 2 years ago, I would have been like laughing at you. But I think we made it. And the team was fantastic at that. But it's like involving a lot of people in the team, bioprocess engineering, all the protein people. Yeah.
Lucile, can you give us some examples of, what are some of the steps that you took to de-risk between moving from the lab to scaling up in manufacturing, and also, how you use the tools at your disposal now to sort of monitor and control that risk?
Yeah, de-risking is probably the right keywords. There is a lot of that into what I'm doing. I mean, when you run R&D in biotech, you have to imagine that a lot of experiments fail and you don't want this to happen at large scale. So you'd rather de-risk properly. The first thing to do is play with parameters. So when you look at precision fermentation, you have a couple of feeds. You look at oxygen rate, you look at aeration, mixing, etc. So you kind of stress test your system. That's the first thing to de-risk, right? You look into, okay, if the feed of glycerol is not working on that specific day, like what's the lower boundary I have and what's the higher boundary so that my cell stays happy and still produce something. So there's all this stress testing happening. Throwing it on the cloud, it is also very useful to guide the manufacturer. And then you also have a stage which is called pilot. So you don't jump from a 1 liter to an actual 10,000 or 10 ton, but you go with 100 liters and maybe 1,000 liters if it's necessary. And you test the robustness of your process. Just to make sure that the transferability is actually true, but also that, the process is robust, that you can repeat it twice in the same condition and nothing changes. So that's the kind of de-risking strategy that you put in place in the beginning.
There was actually this use case where we had anomalous reading from the tanks, and we put the data inside one of the models of the cell we have, and could see that there was something in the metabolism that was not acting as we wanted, and we could act on it at the genetic level and basically prove that, which I think is like perfect proof of concept.
Right. And so that's the feedback loop that you create by having the data infrastructure in place that takes sort of the stress testing that's happening in the lab and brings the data up into the cloud where it can be quickly integrated into the models that you have available to sort of analyze what's happening from a biological perspective. That's really interesting. So I'm imagining that the output of this, sort of exploration of the parameter space of the different variables in the environment or, in the inputs that you expect to vary within your manufacturing process, that the output of that exploration is sort of guideline for your manufacturing partner for, these are the controls that we need in place for how the manufacturing process needs to go. Is that the way that it works? And do those parameters change over time from your perspective? And how do you sort of manage that process?
The thing that you see is that once the parameters are going to change just from an environmental reason, right? Like maybe this day is colder and you have to adapt the temperature differently, but also you have the human factor. when we are working in the lab, we automatize everything. I can write a script on my fermenters and I know it's going to run the way I want because there is this loop system. When you work at higher scale, not everybody is equipped with those kind of technology. So there is also people literally pressing buttons, making decisions at which time, when they see these parameters reaching hundreds, they need to press a button and do it. But there is still like this interpretation of you being like, am I in the range? Do I need to press the button? So this guide is basically helping you to help people make the right decisions and also de-risk it. So you give a very direct guideline, feed glycerol at that time, but if there is 10 minutes difference, in the end, your process is going to go well. So it's exactly a guideline, just very complex to read and I think not accessible to everyone.
How do you approach quality control at that scale as you're scaling up? So, when it's in your lab and you have a leader, it's very easy to sample it and sort of, run your assays against it. But how does quality control work as you're scaling up?
Yeah, maybe one thing to say is that Cambrium is very innovative in everything which is upstream protein design and getting to a process which is very efficient, very fast. When it comes to quality control, there is not so much room to be innovative. You can be much more efficient than everyone else, but there is no space for basically creation. You follow the guidelines. And the key for me is partnering. You just partner with the right people who did that for years, who know the industry, they know the assay, and they can guide you there. So we have a lot of support on consultants, really good partners. And I think there, unfortunately, less improvisation available, but I think you can be creative into driving that the proper way, but you just have to surround yourself with the right people.
Yeah, here the marginal value of domain expertise is so high.
Yeah.
You'd rather bet on the right people.
That makes a lot of sense. So Cambrium is a biomaterials company. You're not just focused on NovaColl, although you want NovaColl to be the first of many biomaterials that you're bringing to market. So I'm just interested in how you think about the development of broader, more integrated software, data science, manufacturing capabilities at Cambrium that allow for that expansion. And, how repeatable do you feel like the process that you've developed is for other biomaterials you might develop versus, additional work that needs to be done in that regard?
I'm going to give a two-part answer. One is Cambrium specific and the other maybe like some ideas I have about the general field. We built the platform so that basically the next molecules we want to bring to market, the price decreases because the platform already exists and is working, right? And we're already seeing results like that in the lab. We're seeing increased efficiencies in molecule classes that are totally different and so on. So for us, that part of the bet is already in good way. There is always this, okay, can you guys do something else than cosmetics question that we need to answer? We have like so much cool stuff that we want to show. But yeah, the product cycle has like 9 years. So hopefully in the next month or years, you'll hear about some of the cool stuff we're preparing everywhere. But I'm super confident about it. I think we proved that it was possible with NovaColl, and now we're gonna bring it to a sort of industrial scale throughout the material world, right? And I think for the market in itself, a lot of movement has been happening on the pharma and healthcare sides of things. I think biomanufacturing is still a little bit the misunderstood child of life sciences in the industry. And here there's like an opportunity for anybody who's a little bit innovating, everybody who's a little bit an entrepreneur to make things happen, right? Like there are things to get done. Biomanufacturing is the solution to many problems of tomorrow. The AI boom that we are living right now is a way we absolutely have to catch in order to make a difference tomorrow. And I would really encourage anybody who's super excited about this to, have it in there, because I think there are an infinite amount of possibilities.
Yeah, there is definitely a lot of space to create a couple of really cool new bioproducts. Beside the pipeline that we are creating, which is super exciting that we can't reveal yet, but you can imagine the protein, everything which is vegan and can offer different properties. I think the space is massive. And I think the data point, which was amazing 1 year ago, was when we realized when we tested the efficacy testing of our first product, that actually it was mirroring what Pierre designed a couple of months before. And if it worked on a substrate, which is as complicated as skin for anti-aging, I'm really excited to see how this goes on different additional applications. So cool stuff cooking in the lab for sure right now.
But really, I want to make space blue. That's my end goal. It's been 20 years, but I want to get this space blue out.
So it sounds like the way that you structured the problem at the beginning, Pierre, and sort of the way the data platform that you've described and sort of the way that you think through the design and scale up of proteins is repeatable. Also what I'm hearing is that what you're saying is that sort of the space for exploration of these different applications of protein design in this mode is wide open. There's a lot of opportunity for innovation there. And also, you don't seem to be particularly scared of competition because there are so many applications of what you're doing. And am I hearing that correctly?
Yeah, I mean, obviously investors don't want to hear that, right? But what one of my good friends said is like, the cake is so big, you can't eat it by yourself. So, bring friends along and build the bioeconomy, right? I do want to echo this. I think there's a lot to be done and there's a lot to collaborate on. We're friends with these people who make molecular cheese for, right? Like, we're not going to make cheese for them and they're not going to make materials for us, right? So, why not help each other? Because at the end of the day, we're both sequencing strains, right? So here there is like an opportunity for exchanges, like building a community together. One thing I want to mention is the space is big, but we have to be smart about how we work it, right? Like when we are trying to make biofuels, we are trying to make commodities with deep tech, which is good when you have backing from the oil and gas companies, way harder when you're a startup. That's why we need to be smart about how we tackle things. You start with things that have a high added value, specialty chemicals, and then you work your way down to commodities. Because ultimately, how are you going to make a material change in the world by replacing bioplastic and concrete and so on, right? But the price points and the quantities for these ones are so high, you can't start there as a startup. So you do specialty chemicals first, and you work your way down the value chain so that you can ultimately change the world. And for that, we need many companies.
Yeah, that's great advice for new startups.
I think we have a lot of really cool profiles in the company who are actually both, who are trained on both sides, because as they come along, they are trained on one side or the other one. That's really the base of creating a team which is going to give you really, really excellent results. I think Pierre and I have two different teams, but in the end of the day, they kind of work together. It could be a single team. So that's one very strong point. And also when you welcome people in the lab, it's like really onboarding them very fast on data and data capture, and be very diligent on why you do that, why it's important, when the quality matters. That's been one of my battle, and also it's very close to my heart. So actually always talk about that, because again, you do R&D, if you do it the old ways, you have no chance to be competitive in the future. And there is no reason to not use those core tools, right? I still don't know how to code, but I can sit beside Pierre and tell him, hey, look, I need that. Do you have a solution for me? And then we build it together. So I think that's a very strong decision that company can make in the beginning when they are creating such team, is also embedding the data journey from very early on.
It sounds like sort of the commitment at the leadership level within Cambrium to collect that data and to make sure that it's driving the decisions that are happening on a day-to-day basis is really important. Are there any challenges that you've faced in trying to do that sort of rapid onboarding in the lab, or trying to make the teams collaborate more effectively together?
Yeah. I mean, outside Cambrium, there is always those people who tell you that I like to do it the whole way. But I think after repeating and showing them that you're actually very fast compared to the rest of the industry and competitive, usually they get convinced. And I think in-house at Cambrium, I mean, you have to imagine people working in the lab. There is a lot of manual work. Everything takes a lot of time. It's very complicated work. You have to be thinking, but at the same time, be very manual. And you add basically an additional constraint of them needing to enter some data in a certain system when they could actually just draft something on a copyboard. So I think like really talking to people that the time they take for data capture is actually part of the job and part of what they need to deliver, and going to save them a lot of time later is actually kind of the endpoint I always mention to motivate people to get behind that. And I don't know, at Cambrium, it feels like the onboarding always has been very smooth and natural, but it's also something we screen for when we are hiring people. Checking the enthusiasm about all those tools is something we also do.
Yeah, and once you have buy-in, it snowballs really fast across the team, because people see, oh, you're saving so much time by doing this.
It's pretty obvious, yeah.
So as we draw to a close, I'm just curious if either of you have any resources that you would point data scientists and biologists toward if they're thinking of exploring, in silico design of proteins, anything out there that you're monitoring that helps people to get up the learning curve of this field.
Absolutely. And here, the first thing I want to say is it's not because you never did biology that you shouldn't try, because it's absolutely captivating and easy to understand, at least at the high level. I would always recommend understanding AlphaFold, not necessarily reading the paper, but understanding how it works, understanding the Facebook ESM embedding model, because it's foundational for a lot of the stuff that's been happening in the protein space in the last years, looking at diffusion models and understanding the power with natural language, right? Proteins are letters, natural language is letters. Composing a protein is the same as composing a poem. If you just randomly choose your letters, you're going to get gibberish, right? But if you do it properly, you get Shakespeare. And that's what this whole field is about. And I think we're really good at Shakespeare and protein speak at Cambrium.
Fantastic. Well, this has been a really insightful conversation and really appreciate both of your time. So Pierre and Lucile, thanks so much for coming on and look forward to connecting down the line.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.